Okay, so meiosis 1 is the first division of meiosis, which separates the homologous pairs. Okay, so it's kind of the same setup as mitosis, where you can see that we have the original parent cell up at the top. Okay, our DNA has been condensed into the individual strands, okay, of chromosomes. So they are single stranded, still 2N. Okay, so this is basically like having the 46 chromosomes. Okay, so then they pair up and become double stranded, still 2N. And that's in prophase 1. Okay, so it's the same exact setup as prophase. It's just called prophase 1 because there's two prophases basically. Okay, so then we go to metaphase 1 where they line up in the middle of the cell, okay, at the metaphase or equatorial plate, still all 2N and double stranded. Then we go to telophase 1. After metaphase 1, we will have anaphase, okay, anaphase 1 where they start separating, okay, same exact setup, and then it's telophase 1 where we will have the two separate cells now. They are still double stranded, okay, because as you can see, instead of just one red, one blue, okay, it's double, it's 2N, okay. So we're still separating, okay, so now we have one red, one blue, one red, one blue in each one rather than double the amount, okay. So now we are at 1N, which is double stranded, okay. Now we have this process called synapsis. That's where it forms the tetrad, which is the 4 connecting. Okay, I'll kind of go into that more later. Now we have meiosis 2. Okay, so meiosis 2 is going to be the second division, which separates the sister chromatids. Okay, so we have our paired chromosomes, okay, and then we have to separate into the individuals, which are called chromatids. This is just like mitosis. Okay, so we go from prophase 2, where they're, all set up in there, to metaphase 2, where we're lining up in the middle. So at the metaphase plate or equatorial plate. Then we go to anaphase 2, where they start separating and pulling apart. So we're pulling our sister chromatids apart. And then we have telophase 2, where we will have 4 individual daughter cells, each with a single stranded chromatid. Okay. Now, the reason this is more complicated is because in meiosis 1, we ended up with 2 cells. So now in meiosis 2, we have this process of meiosis happening in both daughter cells, okay? So in both of those 2 cells, which then gives us 4, okay? So that's how we go from 4N to 2N, or 2N to N, sorry. So here's kind of the steps broken down. So meiosis 1 is the first division of meiosis, which separates the homologous pairs. We go from 2N to 1N. So we are literally reducing and cutting it in half. Then meiosis 2 is to separate the sister chromatids. So we're staying at 1N to 1N. Meiosis 2 is exactly like mitosis. Okay, it just happens in 2 cells at the same time. So if 2N is 48 for a particular cell, then the chromosome number in each cell after meiosis would be, oh my God, you know how to do basic math. Yay, divided in half. Okay, it's 24. So for humans, we go from 46 to 23. So here are real life microscopic pictures of the phases of meiosis 1 and meiosis 2 to compare. Okay, so here you can see for meiosis 1, it's at the top. It's just the one cell that's dividing. And then in meiosis 2, you can see that all of the phases are happening in 2 cells. So then we eventually end in 4. So how, if this happens in everyone, do we get all of this great genetic diversity? We all look so different, and even looking so different from our siblings and our parents. So we have processes throughout this that help create diversity within our genetics. So we kind of trade pieces of DNA. It's called crossing over or recombination. So for this happens during prophase 1. Basically, the sister chromatids intertwine, and the homologous pairs basically swap little chunks of their chromosomes. So the DNA kind of breaks apart and reattaches. So if you look here at metaphase, okay, you can see that the we had red and then we had blue, and then kind of the little chunks of the arm there kind of broke off the red and attached to the blue, and then the chunks of the blue attached to the red. So you can see that those chromosomes kind of broke apart and reattached. So that way we can kind of get this crossing over or recombination of the DNA and genetics. So crossing over happens in 3 steps. Okay. There's a crossover at the chiasma, okay, which is the tetrad. Oh, wait, go back. Okay, so right here, it can be a breakage of DNA with specific proteins, and then it'll be the refusing of the DNA. So here we have our 2 chromosomes, okay. Then here we'll have a crossover at the chiasma, okay, so there's kind of like a tetrad where they kind of end up crossing over. Then what happens is you have the breakage of the DNA and then the refusing of the DNA. So here's where you kind of have it break off and reattach, and then it fuses to where they're separated again. So go ahead, pause, read this question, and then figure out your answer. So your answer should be A, because crossing over creates genetic variation in gametes. Okay, so that's going to be the main question that you are going to be asking. So here's just a side-by-side of mitosis versus meiosis. Obviously, they kind of summarized meiosis 2 pretty quickly, but yeah. Now, that was pictures. Here's words to kind of compare and contrast mitosis versus meiosis. You guys can kind of look that over on your own. So here is a question that they love asking to compare mitosis versus meiosis. So go ahead, pause, read through, but your answer should be D, okay? So mitosis produces 2 identical diploid daughter cells. Meiosis produces 4 haploid daughter cells after 2 rounds, okay? If you didn't understand why others were incorrect, let me know and I can explain it to you one-on-one. So, the value of sexual reproduction, okay? Sexual reproduction is so valuable because there's so much genetic variation within it, okay? So you have the genetic recombination. So the chromosomes get to decide, how they're going to line up and everything and do their own thing. You can have crossing over. You can have random fertilization. So which sperm fertilizes which egg, because each sperm and each egg have their own different genetic diversity. Another valuable aspect of sexual reproduction is that it drives evolution. Okay, if you have all of this genetic diversity, you're getting variation for natural selection. Obviously, certain traits are going to be more valuable in life than others. So all of this genetic diversity is going to increase the rate of natural selection, because you'll get to see so many more traits being more applicable to different organisms. So here's variation from genetic recombination. If you look at the egg from mom and the sperm from dad, okay, you get offspring gets kind of this mix, okay? But then the new gametes made by the offspring itself, look at the different options that you have. Okay, so whatever this offspring is, let's say it's a female, these are the eggs that the female could have. And you can see that it depends on which sperm gets or which egg gets fertilized by the sperm, to where the offspring can have just completely different traits that are more like its grandpa or more like its grandma. So to give you in terms of numbers, there's a random assortment in humans that produces 2 to the 23rd different combinations in gametes. So that means 8,388,608 different combinations in gametes. Your kids can have that many different combinations. That's a lot of freaking options, okay? So this is a Punnett square. We'll get more into Punnett squares shortly, later on in this unit. But this is kind of a Punnett square to show you all of the different options, just, with one baby. So how are sperm produced? Sperm are produced through a process called spermatogenesis, okay? Men continue to produce sperm. Males always are producing sperm until the day that we die, or the day that they die. I ain't a dude. Okay, so each ejaculation produces 100 to 600 million sperm. Okay, so each time a guy ejaculates, they lose that much sperm, but it doesn't really affect them because they keep making more. Okay, this is kind of a visual to show you how the sperm are specifically produced in meiosis, just to show you the picture of the sperm in the end. And then here is a picture of a testicle, okay? And inside the testicle, that's where the sperm is made and stored. And then these are the tubes that transfer it and transport it out to be ejaculated. Egg production for females, you are born with the amount of eggs that you will have the rest of your life. It just, all they do is kind of mature and develop in your body. So that's why females kind of have like a date where, they start to expire. It's called, after you have your menstrual cycle, you'll have menopause. Menopause is basically when your eggs start breaking down because, you've had them since you were born. And so you can't really have children anymore or anything like that. So oogenesis is eggs and ovaries are halted before anaphase 1, because meiosis 1 is completed during maturation, which basically means puberty. Okay, meiosis 2 is completed after your fertilization. Okay, meiosis 2 is triggered basically by the fertilization of the sperm. So next video, we're going to get into errors of meiosis. So we're going to go over chromosomal abnormalities and different disorders that can happen.